This video is going to be about oxidative phosphorylation, which is going to be creating ATP using oxygen. So we haven't really seen oxygen come into play in the first two steps of cellular respiration, glycolysis and Krebs cycle, which are all about breaking the glucose down, releasing electrons, and then carbon dioxide was lost as waste. So where we're at here is this is the outer membrane of the mitochondria. We have the inner membrane space, which just means that it's in between the two membranes. This is the inner membrane of the mitochondria, which is kind of that squiggly line in the middle of our mitochondria. And then we have our matrix, which is going to be the middle of the mitochondria, so that center part. So what we have is we have our electron transport chain, and we have our ATP synthase. So what's going to happen here is all of those electrons that have been released from glucose are actually finally going to make it to the electron transport chain. And how they actually get here is they're carried by the electron carrier NADH. So NADH is actually going to release its electron, and it's going to now, NADH, it's released its electron, and it's going to be oxidized back into NAD+. So NAD+ is going to be what comes out of this, and then NAD+ will actually go back to glycolysis in the Krebs cycle to pick up more electrons to drop off into the chain. So what's going to happen is this electron is going to go down the electron transport chain. But first, we need to note that at the bottom of the electron transport chain is oxygen. Oxygen is going to catch this electron at the bottom of the electron transport chain, and it's going to become water. So what we're going to see is the electron is donated into the ETC by NADH, which becomes NAD+, and that electron is going to slowly work its way down the ETC, and as it does, it's going to pump these H pluses into the intermembrane space. So the intermembrane space has a high concentration and the matrix has a low. So in order to pump them from the matrix into the intermembrane space, that's actually a low H plus to a high H plus. So that is active transport. So in order to actually transport anything low to high, energy has to be used. And in this case, the energy just comes from our lovely electron, which is going to continue down and be added to oxygen. So the electron is added to the oxygen, and that is going to reduce oxygen into our lovely H2O, which is going to be lost as waste. So this is waste product. We breathe out water vapor because we're making it in our mitochondria, and it is not really going to be used. But the most important part of the oxidative phosphorylation isn't necessarily the electron transport chain making water. It's going to be the fact that the ETC does pump H plus from low to high, and it creates this giant wall of H pluses, which can then be used to power the ATP synthase. For ATP synthase to work, it has to have H pluses pass through it in order to make ATP, but they won't pass through unless it is passive transport or it goes from high to low. So the next part of this process is the H plus is actually going to flow from the intermembrane space through ATP synthase down to the matrix. And when it does that, when it goes from high to low, it's going to help ATP synthase make ATP at the end. So actually what we get is as this travels through, ATP synthase is powered up and it makes some ATP. Per glucose molecule, enough electrons are released to create about 24 to 26 ATP, in oxidative phosphorylation. So that's about how much ATP we're getting. And what I mean by enough electrons are released, it's that all of the electrons that are donated to the ETC come from glucose. Glucose is broken down in glycolysis and Krebs, and the NADH comes from those. So enough electrons are released to pump enough H plus up into the inner membrane space to make about 24 to 26 ATP. And that is the last step of cellular respiration, and you can see that it makes a lot more ATP. So this is so important. Without oxygen here to accept the electrons and be reduced, this entire process would not work, because what would happen is if oxygen wasn't present to keep pumping H pluses up into the inner membrane space, eventually this would equal out and reach equilibrium to where these would keep flowing through, and then they wouldn't be being pumped back up. So they would just stay in the matrix instead of getting pumped back up into the inner membrane space like normal. It would reach equilibrium, and that's no good, right? That would mean no ATP, and that's why if we don't breathe, we actually are at risk of dying, because we need that ATP from oxidative phosphorylation to power our cells. So that in a nutshell is going to be the last step of cellular respiration. Very brief recap. Glucose is broken down into carbon dioxide in substrate level phosphorylation. All those electrons that are released when the bonds of the glucose are broken come here. They go through the ETC and reduce oxygen into water at the end. As they go through the ETC, they pump H plus up, which powers ATP synthase, making enough ATP for us to survive. So that is cellular respiration, occurs in the mitochondria, and happens in both animals and plant cells.